semester, the fourth and the last one. And we have today the great pleasure of having Professor Prasanta Subudhi from the LSU Arts Center. He has been in LSU since 2001, and you got the PhD in Italy, you told me. Very nice. And he is an expert on rights, and he will explain to all of us what science that he does. So thank you for coming, and take it away.
But at the beginning, I want to thank all the organizers for inviting me this talk. As you know that last year we got a grant to develop climate resilient rice. So when I got invitation from Dr. Rao, I thought this is the perfect time to highlight what we want to do, what is done, and what can be done to mitigate the climate change with reference to rice. So I have been in rice for almost 30 years, although here 23 years. I have worked in International Rice Institute for 3 years, before that in India also a few years. And most of the time I devoted mostly to rice. So I'm a plant geneticist. So my job is to find variation. What is the genetics behind it? So that is the genetics too. And what is the molecular basis of certain variation? So I will not make this talk very technical because I know there will be diverse audience. So that's why I will look very generic and what has been done in rice, how it impacts the climate change, impacts the rice cultivation, and what has been done or what has been accomplished, or what we can do in the future for getting or design a rice which can help in ensuring increased food production. So this is the climate change in an existential threat for the survival of the human civilization. There is not a single day you might have heard we pass by without reference to climate change. In the news, TV, radio, wherever you go, you will see so much related to climate change, whether it's wildfire, drought, climate disturbances, wind, heavy wind, hurricane. These are all the consequences of climate change. And we discussed about what is climate change. Some people actually, it is synonymously used with global warming, or some people call it a weather disturbances, weather variation, weather fluctuations. Actually, climate change is a very, very broad term. Global warming does not encompass all the things what is happening. Weather, climate, you should know the difference between weather and climate. Weather is like short area, what is happening. That means atmospheric changes, short-term atmospheric changes in a local area. Whereas climate is global in nature and its incidence is almost unprecedented. It is affecting not just US, it is affecting almost all the countries all over the world. You see catastrophic flooding, fire, drought. You see, these are all the consequences of climate change. So, as you know, the world population is growing at a rapid rate, and we can see here that by 2086, we will reach 10.43 billion people on this planet. So we have to feed those people. And for that, the plants mostly actually supply the food. For almost 80% of the food are supplied by the plants. And even if you eat the meat, those are also produced with the help of plant foods. Because animal meat, they also, or chicken, they are also fed by the plant foods. Right. So that is fine, but the good thing is that the population is not increasing at an exponential rate. After 2000, you can see the graph, 2.3% it peaked in 1963. After that, it dropped. Almost half now, 1% rate population is growing. So it will be almost plateaued around 2000 or 2090. So the population is somewhat plateaued at that time. But still then this is a big population. We have to feed, ensure food stability or food security for those people. And what are the impacts of the climate change? This is just, I'm giving you the 2011 and 2020, the global temperature has risen 1.1 degree. And if you see the IPCC report or the climate change committee, they have recommended that we should restrict the temperature below 2 degrees centigrade. We should not allow to increase the temperature, the global temperature, above 2 degrees centigrade or 3.5 degrees Fahrenheit. Otherwise, there will be drastic consequences like drought, flood, all these natural disasters will be more and more frequent, which will threaten our food security and will threaten our survival of the human civilization. Similar with this greenhouse gases, you can see here all these carbon dioxide, methane, and nitrous oxide. Those three are the major contributors to the global temperature. And those are all now, it was around 220 or 240, now it is around 420. Now I think it is 430 ppm carbon dioxide concentration. So it is increasing. So the impact of this, that we are seeing the temperature, global temperature rising. So that will have impact on cultivation of almost every crop, including rice. So what is its impact on the food security? You can see here the left side, the environmental outcomes of the climate change or the consequences of the climate change. To name a few, that flooding, because rising sea level, that will be the main impact, because the glaciers are melting in the pole. So where this water will go? To rise the temperature, rise the water level will increase. Because the water level is increasing, some of the coastal areas will be inundated. And some days it is predicted that like Bangladesh, so that will be underwater. A lot of coastal areas will be underwater. Even there is threat to also Louisiana. We are losing land every year. And every minute I think it will maybe 2 football fields per hour or per 6. So there is some data that we are losing land in the sea. And what are these impacts on the food security? You can see here the impacts on agriculture because of threats of the abiotic stresses like salinity, drought, global temperature, increased temperature, and food stress somewhere, and drought. All these things resulting in disease severity as well as lower yields, and then ultimately it will reduce the quality and yield as well as nutritional quality of every crop, including rice. So why we are working on rice? Because that was my passion was because I grew up in rice with rice, because it was all I have seen in my childhood, all rice fed. So that is why I got interested from the beginning that I will do something on rice. And rice is the most important crop, which is feeding almost half of the world population. 4 billion people depend on rice. And 21% of the protein requirements each supply, 21 of the human energy requirement is coming from rice. And 15% of the protein requirement comes from the rice. So there has been a lot of research. If you have heard about dwarfing rice, because rice sort of plants so that it can give high yield, because earlier the farmers used to grow very tall rice. And in 1960s, when this working rice evolved, and that led to this green revolution. The rice production increased tremendous in 1960s because it was very responsive to fertilizer, and also there was no logic, so that you need to harvest, so you don't have a loss to rice. So that was the time, but now we have to have a second revolution. This is the green revolution for increasing yield. But now, because of climate change, we need a second revolution where we want to stabilize yield. Whatever we have also, we have to have some stability in the yield. Because of fluctuations in the weather, or because of the climate change, we are getting flood, all these types of abiotic stresses. And also natural resources, those are also depleted at a very faster rate. For any crop growth, you know what are the important components needed. You need land, you need water, you need good air. So where if carbon dioxide is more in air, that will have impact on the growth of rice plant or other crop plants also. Yes.
Is there any way that we can make crops resistant to climate change the way we make them resistant to...
That is what we are discussing. We are discussing that. We'll come to that.
Okay.
Yeah, we'll come to that. What we have, I'll talk about that.
Okay.
We are
Thank
just
you.
giving some introduction about those things. Okay. So global crop production is actually, you need the growth. Crop production is, we need a 2.4% growth in, these are the four major crops. Whereas the growth, you can see rice growth is 1.1%. Whereas we need 2.4% growth in every annual growth in rice production. So that without increasing the rice acreage, we can double the crop production, double the rice production by 2050. So that is our goal. And let me talk about the rice, where it is grown. If you are not familiar with that, this is rice, it's the US. It's not a major rice growing country, but it is 11th largest rice producer. But it plays a major role because of export. It is the 5th largest exporter of rice. Most of the rice grown in the US are exported. And all the south regions, you can see is Texas, Missouri, Arkansas, and Mississippi, and Louisiana. Those are the southern regions, and the California as well. Those are two different ecosystems where rice is grown. They are totally, California is totally different from ours. There is different types of, that means environment where the rice is grown. I think you will be glad to know that California, this is the world's rice productivity per acre is the highest in the world. Because the rice, the environment is so conducive there, there is no pest, diseases, all these problems are not there. But the problem is that drought, because for the last 2 years there was a drought, so rice acre is dropped there. Because it is all coming from the glacier, when the, not this snow melting from the hills. So that is what but nowadays I think this year is better. Okay, extreme climate, all these things I have talked about this, heat stress, submersion stress, and drought stress, and salinity stress. Those are the four major things in force stress in some areas. And just the heat stress, you know, every 1 degree rise in temperature, there will be 3.2% reduction in rice yield. So that is an estimate. So let me talk genetics. I will show here that how much diversity is there in rice. You'll be eating all rice, it seems like it's the same. It is long grain or short grain or medium grain. That is only you can distinguish. But there is a lot of differences among the rice. You can see here, in the left, there is a rice, different colors, different sizes, different. And cultivated rice, there are two different types of rice, African rice and Asian rice. We grow it here, Asian rice, which is Oryza sativa, that is the scientific name. Whereas African rice is Oryza verima, which is grown mostly in Africa. And Oryza sativa Asian rice, most of the Asian countries to grow Oryza sativa, it can be further grouped into Indica, Japonica, and Javanica. Those are all the three different types with different characteristics. some are like sticky rice, some are fluffy. So there are different types of rice are there. And even compared to white cultivated rice, we have also a lot of variation in the white rice. So much of variation is there in white rice, and they are treasured for particularly traits mitigating the climate change. Incidence of pests and diseases can be reduced, and also for salinity stress, it can tolerate some of these. You can see here Moraisa Kolkata. This is not very close to, you can easily cross with rice. That is why I made it in three groups: the primary gene pool, secondary gene pool, and the tertiary gene pool. Those who are high school students should know that the primary gene pools, means it can be easily crossed with rice. Even though they are different species, you can make crosses. Whereas secondary gene pool, little bit of difficulty with that. And tertiary gene pool, you cannot make hybrids.
How do you make the crosses?
So crosses, you have to, rice flowers, they have 6 anthers. That is the male portion. So 6 anthers are there. There is a stigma where when the pollen grain falls on that is one fertilized and grain is developed. What we do before the anther comes out, we take it out. So basically we sterilize them. Take out all the 6 stamens, and then whichever desired parent or male parent you want to do, is dust the pollens on that. So that can be done in different ways. For emasculation, that we call it as emasculation or castration, you can say the same word, but emasculation is that. So it can be done also with pump, like dental suction pump. I used to do it dental suction when they suck water from your mouth. I use that. I just cut it, and with the dental that suction pump, I take out the anthers one by one. Then you cross it. You bring the pollen and dust it over there. And then you cover it so that there is no outside pollen there. Otherwise it will be contaminated. So that is what is done. And in some cases where it is not possible, we do embryo rescue. So difficult process, you can do embryo rescue. You cross it, embryo is developed, but that will abort. When it aborts, before that, you have to take the embryo and put it in culture, tissue culture medium, so that it will grow into plant. So those are some of the different ways. But Kolkata is actually, that is a plant which is highly tolerant to salinity, grown in the Sundarban areas of West Bengal, India, Bangladesh, that area. And scientists at ERI actually developed some lines from that, from making process. So, little bit about the genome. If you know about DNA, genome, about this, rice has 12 pairs of chromosomes, means 24 chromosomes. And the rice genome sequence was done in 2000. And there are about 45 to 56,000 genes. And in the genome sizes, you can see here, we are actually way higher. Actually, our genome size cell, human things, around 3,000 MB genome. Whereas rice is having very smaller genome, 380 million bases. You know the base pairs like adenine, guanine, thymine, and cytosine. All these 4 lines which write your genes are written on those 4 letters. So these are all these, means 380 million bases are there in every cell. So developing the climate resilience side. Come to your question. Now, what are needed? We need 4 things. You need appropriate donor germplasm. If you want to improve drought tolerance or high heat tolerance or fire tolerance, so in that case you have to find out a donor for that. And there is a lot of donors there sitting out in the germ bank, gene bank. In ERI, or we have also USDA in US, also donor done for in USDA unit in Stuttgart, in Arkansas. They have also a lot of germplasm there. Idaho also there is a germplasm unit where most of the small grains, including rice, also they maintain. But this is much smaller. But most of the 80,000 to 90,000 germplasm collected from all over the world, they are preserved in the gene bank at Kiri. So that's why anybody can get access to them. I have used some of them from there because in here most of the lines have a very narrow genetic base. And you should know that rice is not native to US. We got it from somewhere else. So it was introduced to US. It is originated in Indochina, and this was a imported crop. And in this case, there is not much variability is there in US. Then second thing is that after the germplasm is dead, so you have to study what are the traits involved in climate resilient traits. So that I will come to what are the different traits controlling those climate resilience in saline tolerance or coding tolerance. Then you have to do the gene discovery. You have to find out the genes and what are responsible for those tolerance traits, and then transfer to your germplasm. Suppose we have a variety, we have developed a lot of varieties which are actually in southern regions. Louisiana is much ahead of other states. Our varieties are the ready, means all over the southern regions. Louisiana Agricultural Center varieties are grown all over the southern regions. Then tools and techniques. What are the tools and techniques you will be using? Plant breeding is well known, that I discussed already, that you make crosses. That is, even the early farmers, they used to make crosses and selection. They are actually the pioneers in breeding. We just have a degree to do breeding, but they didn't have the degree at that time. So they were doing also breeding at that time. So they were the first breeders for any crop. And second thing is the modern genomics tools for the discovery. There are a lot of tools now available, like genome level. We can do sequencing, we can do transcriptome like RNA sequencing. So those are a lot of techniques are there which are much more advanced. I don't want to get into details, but those are tools are there which to find the genes or discover the genes. Then you can use the molecular marker-based tools for transfer of desirable genes. Suppose you find out the genes which is responsible for a certain trait. So, like the PCR markers can be developed, like COVID time. You have the PCR, you develop that whether you have COVID test, whether COVID virus is not there. When we know the markers for certain traits, we can do also PCR-based markers. We can just pinch of leaves, extract the DNA, do the PCR, and test which plant is having that gene, and then use it for further advancement. So that is one thing. And the fourth one, I will come to genome editing, which is a very recent one. I think this one USDA already, it is not transgenic. It is not genome modified GMO. So you should, because some people are confused with genome editing and GMO, but they are not GMO. In medicine, actually, they are used in much more intensively now to cure diseases, because it has the potential to do certain insertions, certain deletions, change the gene. So I'll come to different traits now. Some of these abiotic traits. Submergence of coding tolerance is one, though not it is not in US, it is not very common. You can see here clotting, what will happen in this case in clotting in Philippines, India, Thailand, a lot of places are clotted. Actually, they lose almost lost. I got last week actually one picture from South Carolina. There also they are experiencing clotting. North of Charleston, they are having clotting. All this, this South Carolina, the first variety, the Carolina gold. That variety was grown in there. That is like a heirloom variety. They still love to grow it. There's different types of flavor, different types. But we are collaborating now for developing some projects so that we can develop some clotting tolerance and silent tolerance varieties for them. Because those are not only clotting, also silent, because there is inundation of seawater in that area. So, submergence tolerance is among all the biotic stresses is one of the easiest ones which was solved. It has been worked out that there is a sub1 gene which has been cloned already from A13A, which is chlorine tolerance, chlorine resistant variety. It happens to be this from my state in Odisha, in India. That variety, particularly where the resistant gene they got from sub1 tolerance, from chlorine tolerance variety FR31. And this has been, now you can see the picture, 7-day submergence, that line, how it survives. After 14 days, you inundate with water, but still it survives. And that has been done, you can see that there is a great success story here, and Gates Foundation and other international organizations, they have spent a lot of money for this, transferring the sub1A gene to so many mega varieties, those which are widely grown in South and Southeast Asia. And they have done, you can see clearly the difference where the sub1 has been introduced. These are just pure crossing by hybridization. Regular crossing activity they have transferred. There is no GMO, nothing is involved in that. But he used molecular markers for that. And you can see where there is nothing, no sub1 gene, they are almost dead or they go to stomach. Whereas this is one important success story in the abiotic stress tolerance. Then coming to the salinity, this is a picture from 2005 when there was hurricane in Louisiana. That was a picture taken by one of our colleagues, Dr. Reitenbeck. That salt almost in all these rice areas, coastal areas, there is the salt deposition, you can see here. But after that, it never happened on this much scale. But salinity is also a big problem in the world. Around 25 million hectares suffer from salinity problem. And 20% of the irrigated area are affected by salinity. And rice is somewhat here. You can see here rice 3.0 is the threshold. Decisimen, or you can see electrical conductivity. That is the threshold of rice. It is better than corn and sugarcane, but there are other crops. You see, they are much more tolerant. Like barley is little bit more tolerant to salinity. And in rice, if there is 1 decisimen increase in salinity, there is 12% reduction in rice yield. And the rice has different stages. So it's not all the stages are susceptible to salinity stress. In that case, you can see germination to the flowering or grain ripening. There are 2 stages where the salinity impact so much. It is when the seedling stage or it is in flowering stage. If it is flowering stage, that is salinity stress is there, the pollens become sterile and the flowers become chatty, so that the grains doesn't develop. Whereas somewhat after ripening, the grain is developed, even if there is some salt stress, it can tolerate. So that is why different stages, and most of the water researchers all over the world, seedling stage and flowering stage are mostly researched by the scientists. Because seedling is much more common among the researchers because it's easy to evaluate. The seedling flowering stage actually is very difficult, cumbersome to evaluate for flowering stage. And there are different mechanisms for that. So we have to know the genes for those mechanisms. I'll just come a couple of mechanisms I will tell that ion exclusion. Sodium is one, with the sodium chloride is a salt. So when the sodium uptake is more by the plant, that actually impairs the cellular network machinery to perform its function. And there are others like osmotic tolerance. There are some chemicals or compounds that are produced in response to salinity which can help in tolerating the salt stress, like proline, like glycine butane. Those are some of these osmotic compounds. Another thing is tissue tolerance is there. Plants has the ability also to compartmentalize that salt. They put it in one pocket, in the vacuole. There is a vacuole in the organelle inside the cells. They can transfer the salt to there, so that cells can work, function properly. So those are some of these mechanisms for salt stress. In this case, I will talk about rice because I have been working for almost 10 years on the salt stress. So US germplasm, we found that it's very narrow genetic base. As I told you, it is an introduced crop. You can almost trace their parentage to only 25, 27 grams. That's it. That is not much, means blood from different varieties, or not lot of varieties or germplasm have been used for developing the US vertical. And mostly they are japonica type. Whereas in US, in South and Southeast Asia, most of these grown are indica. Those are different types. And japonica are very less tolerant to this, almost all types of stresses. So in salinity stress, we found out that most of these are indica type. So I got all these, imported all these varieties from Jinban. And the problem is that when you make crosses, they are sterile. Because they, indica japonica, when you cross, even though they are same species, they result in sterility. And that point is very hard to transfer them. You have to manipulate the breeding system, how to transfer those genes. But still then with difficulty, we have transferred those things and made populations and lines developed in that. And most of the lines which you used as donors, they have a lot of undesirable characteristics. You see red pericarp or photosensitivity, it will never come to flowering unless there is short deposition, and also sterility, ons are there, dormancy is there, so many domestication traits. We call it domestication traits. So then once the population is developed, we do the salinity screening. I'll just go through quickly those things. We screen it in like sand culture, put them and different types of stress. We put it for 2 weeks, and gradually we increase the concentration of salt. So after 2 weeks, 3 days, 9 days, and 12 days, after that we score. Different types of scoring mechanism we do. So that's how I'm talking about the breeding work or genetics work, how we do these things. And there are also other traits we evaluate, and salt injury score is easy to evaluate because it gives a very comprehensive picture of the plants. You can give whichever is tolerant, it will stay almost green. There will be no damage to the roots. I'll come to some of the pictures. And you can see here that exotic donors which I imported from outside, you can see most of them are a little bit tolerant based on that salt injury score. Whereas most of our US varieties, they are high scores, means they are very susceptible to salinity stress. And here some of the populations, you can see before stress, and this is after stress. You can see still there are some plants which are green in nature. So that means you can screen and identify those which are tolerant plants. And coming to this data, don't worry about those data, because this score you see, Monocultura, this is one of the lines, donor lines we use. This score is lower score is better score. And Jupiter is one of the varieties released by our action, LSU action. This is very high score. So that means it's very highly susceptible. And these are some of the lines we developed which are close to like high degree of tolerance. It's not like the donor, but it's comparatively high degree of tolerance. You can see here also some of these lines we developed compared to these parents. The parents are dying after, and those are tolerant donors. And some of these lines we developed which are tolerant to salt stress. And also, from those tolerance, we do the genetic analysis, and we found out how it's done. I don't want to get into depth, but those are some of the genes which are identified, which are responsible for salt stress tolerance. These are just crossing between donor and our LSU release varieties. So we realized that it's not that getting genes from one donor may not be sufficient. That's why I ranked these all the genotypes. These are all imported donors, and you can see here those traits. None of this single variety has all the traits. They rank differently. So that means not a single germplasm having all the desirable traits. You can see if your salt cis score, salt injury score. Vocal is one of the best among all the donors. Whereas sodium potassium ratio, vocal is at the end. So that is sodium uptake, potassium uptake, that's called sodium potassium homeostasis. In this case, that is also another important criteria. So that means it performs for some traits better, but some of the traits, other varieties are better. So that is why we realized that you have to get genes from different donors. Make it more so that more tolerant. If you want to develop more tolerant, you have to interface or transfer the genes from different donors. So with that in mind, we made some crosses. And on these blue lines, you can see the blue rounds, those are elusive variety, whereas this red and purple one, those are variety. It's in complex crosses we made and developed some populations with two, sometimes three donors. Developing populations with the objective of pyramiding some of the genes for salt tolerance. And you can see here in the screening in the greenhouse, you can clearly see there is segregation for salt tolerance and tolerant ones, tolerant and susceptible ones. And in one of these lines, you can see here, it is almost like parents. After this phase, you can see it's almost staying green, like stay green type after 2 weeks, 3 weeks of stress. So some of these lines we already multiplied, and they have starting from making process when it becomes sterile. Now at the end we have developed some of the variety which are at working plant stature. That is what we could farmers want. And the maturity same as the parent, LSU accent of parent, and good seed fertility, and no undesirable traits like photosensitivity, red cornel, or ONS, or other traits. So these are some of these varietal trials. I don't want to go into details. You can see one of the lines which is scored also better than the shapes. So 2-3 years we evaluated them, and these are some of these populations lines developed from multiple parents, using multiple parents. So that is also doing good performance at the 5th edition. And what I told before, all the salt tolerance, most of them are seedling stage tolerance. Flowering stage tolerance, actually it is very difficult to, as I told before, is very difficult to screen in the greenhouse condition. That is why the most important thing is that you should have a testing site. Right. I got this from International Rice Research Institute just 2 weeks ago. They have found a place closer to the sea, 200 meters from the sea, and they are evaluating there. You can see there are actually multiple parents were involved in developing lines. Some of the lines, they are really performing well. And they are standing straight, and all these are susceptible lines that they almost. So those are all flowering stage tolerance lines. So those are some of the accomplishments done because Rice Research Institute, they are exclusively for rice research with a lot of funding. So they are able to do that in multiple states and multiple countries also. They are doing. Coming to drought stress, and here drought stress means low moisture content. And there are 3 different mechanisms: avoidance, tolerance, and escape. Everything is controlled by different traits. I'll just briefly tell about avoidance. Deep roots. When the rice is having deep roots, it can avoid the drought stress. In tolerance, osmotic adjustment, like they told in salt trait, many of these actual traits, abiotic stress, they are very common in nature, even the salt stress and drought stress, because in salt stress, when there is first exposure of the saline or salt water, it actually induces drought stress, osmotic stress. First shock is osmotic stress. And after that, when the salt is coming, then becomes ionic stress. Sodium, potassium, so when sodium increases. So that's, in that case, is osmotic fast, then it becomes ionic next. Ionic stress because of sodium accumulation or sodium uptake. But in this case, it's mostly osmotic stress, and deep root can take water from the deeper soil layers. Tolerance and some of the escape mechanisms, like you breed for the early flowering, early maturity type, those type of things, some of the traits you can use also. And it's by 2025, there is one estimate that drought will affect almost 15 to 20 million hectares by 2025. So this will be a major work. This is also another work from ERI. They have introduced both drought tolerancy and submergence tolerancy. The flooding tolerancy I talked about in the first place, sub-1 gene. So here there is the screening of for the flooding tolerance, whereas this is, you see the soil is cracking there, so there are drought tolerance. They combine all the genes to develop those drought and flooding tolerance. There are many genes also have been cloned, and DRO1, one of them is actually the deep rooting gene, which is already known. That can be introduced to the rice varieties to avoid drought stress. And SNAP1, that is also another important gene, is enhances spike protein. Okay, this is about drought. I'll come to now heat stress. Because you are feeling heat everywhere, now all over the world, that heat is becoming a major issue. So, just 2023, it is around, compared to other years, it is the hottest year. 1.78 degrees centigrade more than the other years. So that is impacting crop cultivation, in rice cultivation particularly. And in rice, there has been studied that daytime temperature is not that important, but high nighttime temperature, if high nighttime temperature is high, so that impacts so much. And you know, all the rice, majority of the rice grown in the US for export purpose. So when the high night temperature increases, it results in chalky grains. Everyone knows about chalky grains. What are the chalky grains? Because rice is almost like, it should be like transparent, like glass. But sometimes white spots, white belly, you can see those. Those are chalky. That means the starch is not deposited, packed properly. So that is why they are loosely. They are loosely packed. It becomes white. That reduces the quality of the rice. That is why the export market, actually they do not like it. Because those which are Middle East countries, or we are exporting to Latin Americans, most of the rice grown going to those countries, they don't like it. Because in South America, like Uruguay, Paraguay, the quality of rice is very good compared to US. So that is why the high light temperature tolerance is also important. And heat stress can affect all the three stages. So it can be vegetative stage, reproductive stage, and killing stage. I've listed here what are the impacts of those things. If heat stress impact the killing stage, the reproductive stage, they can reduce the yield drastically. Because it reduces spikelet number, it can be pollen viability is reduced. So it will result in actually chalky grains and reduce tremendously the rice yield and quality. But the good news is that there are some varieties which are good in this. There is a lot of research in one of the varieties. This is Nagina 22, it's also from India. That is one of the varieties most widely researched for heat tolerance. And this is the early maturing offline variety, very high quality, means not much chop, and even in high temperature. So that is why our hypothesis is that that means grain filling rate, if the grain filling rate we can increase it quickly, if you can fill the grain, then we can reduce the choppiness in the grains. So that is what this variety does, and we have been using this as a variety. And you can see here the performance of the N22 compared to other varieties. and there is high milling rate and high head rise, and also chokiness is very negligible compared to one of the standard variety. And even in high stress, A22 you can see here the pollen grains, there are controlled, but still then there is a lot of pollen grains are moving. That means pollens germinate on the stigmas of these rice. Okay, with that actually, these are all the four stresses I discussed about that. So ultimately take home a message is that it is not the yield, just productivity or enhancing productivity is not important. It's also equally important that you stabilize the yield because of weather fluctuation, climate change, all these things. It is necessary for, it's like an insurance. Like you can stabilize the yield if the genes for climate resilient traits is there. Control condition is okay, but if there is no stress, it will still should yield well. But whenever there is a stress, then it should also perform better. So that means we should stabilize our yield. Second thing is designing will require pyramiding of genes. As I discussed in salt tolerance test, I talked about four different stresses. Submergence tolerance or clotting tolerance is one of the very simplest traits because they will just transfer only one gene and it improved the tolerance to chlorine. Whereas drought stress or salinity stress or heat stress, these are not so simple. There are multiple mechanisms involved. That means so many genes are there, they are very complex in inheritance. It is very difficult to manipulate those genes. So that is why you have to have genes from different varieties. You have to pyramid those genes, stack those genes in varieties so that it can improve tolerance. Third thing is wild species. I talked about wild species. One of the wild species, Oryza nivara, that is a wild species. The virus or grassy-stump virus, those genes have been transferred the tolerance to those viruses. And many of these wild species, they are also tolerant to climate resilient, climate resilience. That means many of these amino stresses it can tolerate. So that is why, but it is difficult. But use of molecular marker assisted selection also, I talked about that use molecular markers. Instead of evaluating them again for screening for the ideal or the desirable types, sometimes at the seedling stage, we can evaluate them with just take the leaf samples and select for which one is having that particular gene. So that can reduce the time of developing the variety. It can speed the selection. It can speed up the developmental process, variety development process. We'll come to the challenges and opportunity. I will discuss that these 5 challenges and how to address them. Number one is transfer. I talked about that we need to transfer those genes from wild species and land races, which are but many of them are cross incompatible, results in sterility. That means it doesn't set seeds. In that case, we have to have like careful breeding manipulations. That is, plant breeders have to find ways how to make those things work. Sometimes we reverse the direction of the process. There are some things based on experience. So because all these populations also, I have developed some of the populations, even though they are sterile. So that can be manipulated. And then use of genome editing, we have not done much, but that can be done if you find out some genes. So it can be changed in that particular gene you can manipulate. Second thing is molecular and physiological mechanisms. That is important because if you identify the genes, it is easier to select for plant and develop. The speeding, the varietal development process can be faster. It can accelerate the breeding process in developing those varieties. So in that case, use of modern genetics like genomics, transcriptomics, lipidomics, all this technology is there to find out the genes for different traits which are responsible for climate resilience. Third thing is pyramid. And pyramiding of climate resilient traits is cumbersome because when you develop those pyramidal lines, you have to do multiple processes. and you have to use molecular breeding. As I told you, the PCR-based markers, if some of the traits you know, so that can speed up. Because if you make the process, there will be a lot of genes combinations will be there. So in that case, if you have found out the genes, then it will be easier, much easier to select different, even 2-3 genes at a time you can select. And most of them have been easier, because these are difficult. But we have done it in disease resistance, like in rice blast resistance, rice blast or rice bacterial blight. Those genes have been pyramided. It has been much easier in disease resistance. It has been successful. We can pyramid. We have pyramided the genes. But here it's not yet, because these abiotic stresses, there is a lot of crosstalk between different stresses and very complex mechanisms and interaction with the environment. Evaluation is mostly in common controlled greenhouse environment. You see, most of the publications, that is also one of the things usually people complain or criticize, that what the researchers are doing or molecular biologists are doing, they evaluate in just in greenhouse conditions, lab conditions, some of the petri dishes, like Arab.6 research, they do it. So that doesn't help. So that cannot be translated into field conditions. So that is why we have to focus on the field condition. Whatever you develop is lab condition or greenhouse condition evaluation does not always translate into the varieties which can withstand in the field condition. So that is why you have to have selection, good selection site, good phenomenon is important. Okay. And then fifth thing about the public. Public is very skeptical about the climate change. Public, they are not very receptive. You know, GMO, they are not receptive. But even the new technology, when it comes up, then they are not very, they will be suspicious. So that is why for that there should be awareness. We have to increase the awareness, and especially the farmers, like efficient, we have in agriculture system, like we have teaching, research and extension component, like dealing with the farmers, our extension personnel, they are dealing with farmers. They should be responsible for effective translation of technology and increasing the awareness among the farmers that yes, this technology is good, and this is not GMO, this is what you should grow there. So that's the popularize the technology. Thank you for listening.
So if you have any question, go ahead.
So when you transfer genes, are you doing it with CRISPR rather than just doing crossbreeding?
No.
You're not using
we don't
CRISPR?
do CRISPR.
Wouldn't that be more efficient?
Well, CRISPR, you need. That is CRISPR-Cas9 is what I call genome editing. So that is the technology. And CRISPR-Cas9, you can do that, but for that you have to know the genes.
But
You have to know first the genes, some of the genes which are there, and then you can manipulate those genes. Suppose we have because gene is having different forms. Suppose I have a donor which is tolerant to drug, whereas another line which is susceptible. There is a gene A, which is having 200 or 1000 base pairs, here it is 900 base pairs. So some 100 base pairs is missing. So CRISPR can do that. That 100 base pairs we can change it, we can insert it. But the prerequisite is that we have to know the genes first. And CRISPR-Cas till then, it's not yet very common in everywhere. We have to have skilled personnel to do this. Yeah. And farmers are not yet receptive. Rice farmers are very skeptical. We have to be very careful. Even I worked on transgenic for some time. I stopped it because I was dealing with the farmers. So because if there is contamination or some lines, then it will be a big issue.
so once you know the genes, so I see how that's a problem, but wouldn't that be the most efficient way to get them into a resistant variety?
Well, you can do that if the gene is known. Why to do CRISPR? I can do it by crossbreeding. Only thing is that it will take more time. If I know the donor which is having that gene, I can make the process and select for that gene using PCR. That can be done, but you have to do several back crossing. You have to cross it and select, cross it and select, cross it, select. So that your donor, the recipient parent, suppose we have a variety Jupiter. So that Jupiter, we want to include the gene A. So you should be like Jupiter, but with the gene A. So for that, we have to make several back process and select at each generation for that particular gene.
But CRISPR would be a lot faster.
Yes, yes, it will be faster. It's next generation, but you have to form tissue culture and transformation and then select for that. Yes.
So with like cross-pollination, if you have something in front of that's like if it gets contaminated with another kind of pollen that's not the target pollen, is there a way to like decontaminate that, or do you always have to completely start over?
You have to start over. You never know what is there. Which is that means the pollen comes from which the donor. You never know what will be the impact in the next following generation. Actually, sometimes that is my experience is that suppose varieties. Rice is self-pollinated. You know that rice is self-pollinated, not like maize. Maize is cross-pollinated or sorghum is cross-pollinated. So rice is cross-pollinated means the pollen from the stamen falls on the same stigma. Okay? So it doesn't go outside that much. But there is some amount of cross-pollination occurs. So even though variety is bred, suppose one variety is there, after 20 years growth, you will find that there is maybe little bit of here and there, being sorted from there. That may be due to cross-pollination.
More questions? I was wondering how this crossing modify the nutrient content of rice.
Nutrient content?
Nutrient.
There is already success in our LSU Ag Center. We have developed high protein rice.
Like that.
It's there. It's already not modified by CRISPR-Cas or something. It was by mutation. It's already there, and also glycemic index, low glycemic index rice is developed also in rice. And for my case, I am now working one of my pet projects to develop high anthocyanin rice. Because you know anthocyanin is antioxidants. I want to develop that black rice. I am now transferring the traits from other which are black, so that I want to make it much more black, so that it can increase the anthocyanin content or antioxidants. So those are some of the like nutrients. Because black rice, you know, that is called forbidden rice in China. That only the emperors were using it. Only those are accessible to the emperors. But now it will be ready. I'm still working on that.
Work on it, please. Any other questions? So before we leave, let me just to emphasize that this is the last talk of the semester. We will resume it next fall in September. We will have one talk per month, one in September, one in October, November, and early December. I hope I will see you here during the fall. We are thinking about exciting topics and speakers to come. So I anticipate it's going to be a fascinating program. And before we leave, because you see me here, but this is not run by me, it's essentially run by grad students in LSU. So I would like to ask the students to stand up so we can ask the public to help me. Thank you for your contribution, and thank everybody for coming. I wish you a good summer. I'll see you in the fall.
So one thing, how many high schools are involved here? How many different